Aging is all the rage. We're constantly promised that we're on the cutting edge of really understanding and outsmarting aging.
Our mathematical and numerical analyses seem to show cases where aging would come to a stop. And we too found the same results. Only now, unlike virtually everybody else, we were quite convinced that it was not an artifact. We were convinced that it was always there as an undiscovered corollary of the evolutionary theory of aging. We then embarked on a series of experiments, and they did indeed stop aging.
You've used the magical computational power to do something no one thought was possible. The breakthrough, you guys actually carried it out.
In the fall of 1977, I realized there was this specific term in one of the key equations to produce at least a doubling of average lifespan and a doubling of maximum lifespan.
That would be the equivalent of seeing a human surviving into their 240s.
So, say 200s, 220s.
Wow.
Yeah.
Though I started with little faith in the prospect for understanding, much less controlling aging, my years of work have accumulated into a case for the feasibility of postponing human aging, slowly but substantially, starting from an evolutionary foundation. One of the ubiquitous dreams of mankind, the conquest of our aging, is now foreseeable. It hasn't arrived yet, but a juggernaut has begun to roll forward, and it is picking up speed. I doubt the scientific campaign to increase the time span of human health can be stopped. Slowed, yes, it could be, but a long tomorrow is coming, inevitably. These are the closing words of the preface of The Long Tomorrow by Dr. Michael Rose. A hallmark of the biomedical research into aging and lifespan has been a focus on the cellular and mechanical damage accumulated by living organisms. Many prominent researchers in the field have hypothesized that it is this inexorable decay that leads to aging, but one researcher has spent the last half-century contending that this is a fundamental misunderstanding of aging and forging a new understanding of lifespan extension through both mathematical theory and experimental demonstration. Today, we have the privilege of hosting Dr. Michael Rose, a pioneering force in the field of evolutionary biology and one of the most important contributors in the experimental genetics of aging. Dr. Rose serves as Distinguished Professor at the University of California, Irvine, where he has spearheaded groundbreaking research that has reshaped our perception of the factors underlying the human lifespan. Welcome, Dr. Rose. Thank you so much for joining us tonight.
Thank you. It's a pleasure to be here.
Aging is all the rage. We hear nonstop about the modern obsession with the science, the medicine, the aesthetics of aging. And we're constantly promised that we're on the cutting edge of really understanding and outsmarting aging. Before we can tackle any of that, can you tell us how you define aging?
I define aging at the start of my 1991 book, Evolutionary Biology of Aging, as a period of progressive deterioration of age-specific components of fitness, think survival and reproduction, separately from any deterioration in the environment or any type of infection. In 1991, I thought that process continued through adulthood all the way to death. I no longer believe that. So in 2012, with a group of other authors, we published a discussion article with rejoinders, responses, called What is Aging? And I believe that's open source access. So if you wanted to include a link in the show notes, that would be great. I think it was in Frontiers in Genetics.
Can you build that out for us some more?
Yes, I could happily talk about just that one point for an hour. In 1939, huge amounts of data from human demography were analyzed by two researchers, Greenwood and Irwin, and their analysis seemed to show that aging stops in Europeans living primarily in the 19th and early 20th century at about age 90. Then in 1992, two colleagues of mine and their labs, Jim Kurtzinger and Jim Carey at University of Minnesota and UC Davis, respectively, published really massive insect demography studies, in their case under very carefully controlled laboratory conditions. The 1939 publication convinced no one, not even its authors, that aging stopped. The 1992 publications convinced its authors that aging appeared to stop, but like the 1939 authors, they said, no, but that can't be true. It must, in fact, continue in some way that's obscured by something like some type of weird heterogeneity in the cohorts. Jim Kurtzinger's lab then spent another 8 years demonstrating, actually, no, it doesn't look like heterogeneity in the cohorts or any other artifact is a plausible explanation for what we're seeing demographically in large numbers of laboratory insects that are very carefully handled. During the mid-90s, my colleague Larry Muller and I started to work on mathematical theory derived from evolutionary genetics as to why aging might ever stop in animals or plants in which it starts. And our mathematical and numerical analyses seem to show not only were there cases where aging would come to a stop, it was like really easy to find conditions in which aging would stop according to evolutionary genetics. So then we too joined the party started by Kurtzinger and Carey, and we began to do experiments in the late 1990s on whether or not age-specific rates of survival stopped declining later in life. And we too found the same results that had been repeatedly discovered before. Only now, unlike virtually everybody else who had discovered that phenomenon demographically, we were quite convinced that it was not an artifact. We were convinced that it was always there as an undiscovered corollary of the evolutionary theory of aging, which my whole career has been based on. We then embarked on a series of experiments that only stopped in the 20-teens, where we took our theory through its paces. We extended it to fecundity. We extended it to virility. We extended it to a diversity of physiological characters. And what we showed over and over again, so long as you did an appropriate assay on this diversity of functionally aging characters, that they did indeed stop aging later in life. And the data are voluminous. Some of them are collected together with a theory that Larry Muller and I developed in a book called Does Aging Stop? ? Authors Muller, Rausser, and Rose, published in 2011. 20 years after I published the book Evolutionary Biology of Aging, which was ironically almost immediately made obsolete. The full story of the background to my work and my work itself, together with all of the colleagues I've worked with, whether directly or indirectly, has just been published this year in a book called Conceptual Breakthroughs in the Evolutionary Biology of Aging, which I think supersedes all of my earlier books on aging.
Which I'm greatly looking forward to reading. Now, you mentioned in the book, Does Aging Stop? You talk about this third phase of life that we essentially have a climb up the hill to reproductive maturity. And then the force of natural selection kind of stops caring about us. And we kind of go into this decline where mortality at the population level is increasing. And then, of course, the discovery of this third phase of life, this plateau, I think is of interest to everybody. It would be of particular interest to all of us if we could reach that plateau sometime before we are 90 years old. And that's kind of the holy grail of true targeted anti-aging. But I'm fascinated by the split in your career. You've approached aging from seemingly two different timescales. And I think this is something that's very easily missed when people first encounter the evolutionary biology of aging, because there's kind of this long distance moving target where you've described these extremely long term, super multi generational experiments that you have become world famous for in your laboratory, where you've essentially used the evolutionary population genetics to drive these extended lifespans over multiple generations. And then you've done something completely different with the same model organism to discuss how we can make gains in longevity in a single generation based on lifestyle factors. Can you start by describing this sort of long-term approach where you've almost used the magical computational power of evolution to do something no one thought was possible? And then we can perhaps go down the route of what can we do in a single generation that we could perhaps apply to human medicine more directly.
Okay, sure. So in 1975, I get dragged into the field, kicking and screaming and protesting by my doctoral advisors, Brian Charlesworth and John Maynard Smith at the University of Sussex.
And for our non-biologists listening, these are two of the statesmen of evolutionary population genetics. I can't imagine studying directly under Maynard Smith and Charlesworth, but yes. So kicking and screaming, the field of aging wasn't what it was at this time, I take it?
1975, the field of aging was in notable disrepute. I didn't want to work on the problem of aging. It took them more than a year to persuade me to do it. Fortunately, most of that year was before I arrived at the University of Sussex. So within about 2 or 3 months of persuasion, no brass knuckles were used, mostly mathematics. I was finally convinced that there was a useful evolutionary theory of aging and that I should do my PhD on it. And they wanted me to basically do the experiments that would show that the theory was cogent and the details of how it worked. And I think I actually made a fair start on that project with my doctoral thesis. The first publication was in this obscure journal called Nature in 1980. That's an academic joke.
Very obscure, for sure.
for those of you who aren't academics, that's the #1 journal in science.
Yep.
So, and in fact, Charlesworth and Maynard Smith had given me a wonderful start to my career, and it's been absolutely a charmed life in the academic world. I've been doing research for 50 years. I started in 73. I'm retiring sometime in the next 12 months. And it's been glorious. And the key moment that really, I think, made my doctorate successful was in the fall of 1977, when I realized there was this specific term in one of the key equations for the evolution of aging, which I could in fact tune in the laboratory. And if I move this specific thing, which was the first stage of reproduction in a population's evolution, not when you first get lucky in high school or college, but in the entire population, and then having changed that parameter, let the population evolve for generation after generation, which could be as little as 15 generations or as many as 1,000 generations, and we've done both. I expected that the theory implied that I should be able to produce evolutionary genetic extensions to lifespan, evolutionary genetic reductions to lifespan.
For an experiment in sort of the setting of something like a cell culture, if we're testing something like an enzyme with direct mechanisms, these are things that we can essentially test in real time. The breakthrough in research of this kind, in showing that the mathematics of evolutionary population genetics works, you guys actually carried it out over so many generations that you got to directly measure a shift that to bring it home for those of us who are taught to think in human lifespans. Tell us what kind of goalpost shifting this would be in terms of age of first reproduction and lifespan. Like, how scale this for us in human terms. What kind of shift would this look like had this been some massive human experiment? What would that look like?
Right. So basically, not right away, but progressively over generations, we shifted the first age of reproduction of fruit flies from 14 days, which is sort of be like 21 years of age in humans. Like, it's not terribly early into adulthood for a fruit fly to reproduce at 14 days of age at 25 degrees Celsius, to 70 days of age from egg. So that's 5 times later. That would be like shifting progressively the first age of reproduction in a human population to 100 years of age.
Wow.
So that was the intervention. And the result of doing that is at least a doubling of average lifespan and a doubling of maximum lifespan in a finite cohort. And
so for humans with a maximum lifespan, we obviously have not attained a 120 year average lifespan, but we have observed humans living up to around the 120s.
Yeah.
That would be the equivalent of seeing a human population where the most elderly individuals are surviving into their 240s. Are we scaling that right?
So we see, yes, say 200s, 210s, 220s.
Wow.
Yeah.
And then the intervention was not that you guys made them live this long by moving the lifespan. What you did was you moved the age of first reproduction. How did you go about doing that? And can you explain the evolutionary mathematics for why it was expected to have any impact at all? I think so often we think of single generation when we're not taught to think of things in the accumulated, contingent, adaptive, evolutionary paradigm. We tend to think of things in single generations. And in a single generation, if a couple decides they're going to have their first child when they're in their 40s, they still only get to live in their one generation to whatever expected lifespan they would typically fall into. So say 70s or 80s in a Western country. How is it that the accumulated effects on the population add up over those generations? What's the theory behind it, and how does that work?
The evolutionary theory of aging is that aging is nothing more nor less than the age-specific decline in fitness characters arising from the age-specific decline in the intensity of natural selection at each age in the evolutionary history of a population. Nothing else. So whatever physiological mechanisms are responsible for your strength, your speed, your intelligence, your visual acuity, whatever adaptive attribute you have, you only have that because of the force of natural selection acting on the evolution of humans who are your ancestors.
Up until what stage of life?
And therein lies the rub. We shouldn't be fooled by recent demography. The evidence is that with the advent of agriculture, human health went to hell, and literally human skeletons started to fall apart as people stopped eating the very high quality, if not lower quantity, hunter-gatherer diet. and shifted to eating monotonous diets with large amounts of calories every day, but much lower quality nutrition. It's controversial how long hunter-gatherers could live before they started agriculture. What's not controversial is that if you give humans who have only hunter-gatherer ancestry a diet of agricultural foods, as happened to the Pima Indians in the American West in historically recorded time, their health goes to hell. And they develop early onset type 2 diabetes, heart disease, obesity. It's ugly. Conversely, if you take those same hunter-gatherer populations and give them some type of approximation to a hunter-gatherer diet, as has been done in very recent times in Australasia, you get a massive recovery in health. There's a lot about human demography before the 20th century, which may be more pessimistic about our lifespans and our aging than the actual raw numbers suggest. On the other hand, we have in the 20th century had to have excellent access to antiseptic medicine, later vaccination, antibiotics, and even antivirals, which have saved hundreds of millions of lives and greatly extended our average lifespan. So it's very confusing to interpret the human pattern of aging in the face of both these very big environmental lifestyle upheavals over the last 20,000 years, but also the undoubtedly rapid genetic changes that have occurred in response to agriculture. And almost certainly young people are well adapted to agriculture, providing they have Eurasian ancestry. Everywhere else it's less, but in Eurasia, probably most young people 30 or under are well adapted to agricultural foods, especially if they are supplemented with some hunter-gatherer type nutrients like fish, some animal tissue, fruit, some vegetables.
The idea with the ability of natural selection to derive these genetic changes explains why over the course of, say, 10 or 12,000 years of ancestral exposure to agricultural nutrient sources, we can undergo a shift, a selective shift that at least during early life phases, when we're in our reproductive years, allows us to tolerate to some extent with limited chronic disease burden. Help us understand the connection between why we aren't adapted to simply keep on living indefinitely. Why doesn't the sort of common conception or perhaps misconception of fitness get maximized? Why is reproductive fitness different from the kind of single individual fitness that would allow us to keep living forever on the kind of agricultural diets that we're seeing?
The speed of adaptation to an environmental change is scaled by the same forces of natural selection that give rise to aging in the first place.
Yeah, you got to definitely open that one up for the audience.
So let me unpack what was a verbal summary of some math. Let me give you a metaphor, one I'm fond of. As a child, I was kind of fascinated by fire engine ladder trucks, with the fast ladder thing that followed behind the main part of the fire engine. Because of course, how do they get around corners? And the answer is, of course, they can't get around corners very easily. In some versions of those fire engines, they have a second driver at the back who's furiously turning a steering wheel, trying to make the ladder go through the corners as adroitly as the front part of the fire engine can. So natural selection is kind of like that. At early ages, natural selection maneuvers pretty fast and fairly rapidly adapts populations to changes in selection regimes, like the wholesale dietary and lifestyle change of switching from being a hunter-gatherer to being an agriculturalist. That's why young people, certainly under the age of 30, can flourish literally on pizza Maybe not Red Bull and vodka, but certainly on pizza. And then after a certain age, that really fades away. We have firstly studied this mathematically, again with Larry Muller, and then we even studied that experimentally because without intending to, we imposed a Neolithic revolution on our fruit flies, where we took them from a rotten apple-based diet, which they'd had for centuries, to a simulated rotten banana diet, which we gave them in the lab for hundreds of generations. So we had inadvertently simulated the human Neolithic or agricultural revolution in our lab. And lo and behold, the flies did really well on banana food.
That was the equivalent of representing the human agricultural revolution. This is the shift to a newer, perhaps more available, easy to access type of high calorie food.
Well, it was simply the diet that we adopted in the summer of 1981 when I moved to Dalhousie University, my first faculty position. And the flies adapted really well to that at early ages, or more precisely, up until the first age of reproduction and how I cultured them. So the populations that were kept at an early age of first reproduction, they were superior at that, the banana diet, only when they were young. And then they progressively lost that advantage and went into ill health later in life compared to being on an apple diet. And this wasn't due to idiosyncrasies of apple versus banana. The populations that we reproduced at much later ages of reproduction, first reproduction, they sustained adaptation to banana diets much, much longer. And very, very late in life, started to do better on their ancestral apple diets. Of course, with the advent of agriculture, our generation started to become pretty damn short because we did not live very long. We had pretty nasty, brutish, and short lives.
I think that's a recognition that has become more and more insightful when we think about the kind of dietary shifts that have made their way into the genome, because it was for some time imagined that hunter-gatherer lives were the short and painful lives that were then enriched with agriculture. And of course, as you have mentioned, it really was quite the opposite. The chronic disease burden to this day in hunter-gatherer populations is now known to be exceptionally low, to the extent that it's nearly untraceable for some diseases that are primary disease prevalence in Western countries with access to unlimited agricultural, post-agricultural, and very, very processed foods. We would expect across species, from the model organisms that you're dealing with in the lab, like fruit flies, all the way up to humans, this mathematically consistent prediction that the effects of natural selection are limited to this reproductive window, because the term antagonistic pleiotropy.
I have to stop you. It's actually worse than that. That the full power of natural selection is in the pre-reproductive phases and the very early reproductive phases. Very soon.
So if you have a gene that leads to death prior to your first reproduction, then you pay the ultimate cost of being eliminated from reproducing in the future. You're out of the gene pool. That makes total sense. On day one, we're born. We have no offspring. Natural selection is absolutely brutally fully enforced. Anything that can impact us and prevent reproduction or prevent survival before reproduction, then first reproduction happens or second reproduction happens. And then at this point, let's say for our listeners who are older and are done having children, the curve begins to drop off. The power of natural selection. Why does that open the window? Why can't we say, let's take these healthy, strong, reproducing bodies that we have and keep on living? Why is that not a part of the prediction?
Okay. The way evolution by natural selection, acting on genetic variation, operates is like economics, like physics. It is mathematical, quantitative, and brutal. It is not verbal, intuitive, or pleasant. That's the way evolution in natural populations works. It's just like if you have a market, you therefore have some of the strictures of market economics, and you get supply and demand. You get the top 20% taking most stuff and the bottom 80% being screwed to some lesser or greater degree. It's just like you can't go faster than the speed of light if you're a physical object in this universe. That's just the way the math works.
Within genetics, we have lots of almost terrifying examples of this. For example, the way that a fetus is able to survive in the setting of a maternal immune system is that it expresses these exapted viral proteins, these syncytin proteins, and the endogenous retroviruses that are in our genome that code for the exact protein that protects a fetus from the maternal immune system. In later life, after serving this really, really important purpose of keeping a fetus protected, helping it survive, in later life, that same individual can go on to develop a host of autoimmune diseases, including some like multiple sclerosis, due to accidentally re-expressing the exact same endogenous retrovirus derived envelope protein. These are the kinds of payouts that are wonderful. They're a benefit to survival in early life, but they're a cost in later life. And this idea of antagonistic pleiotropy that you've spoken so many times about explains so much of the kind of disease burden that we see in old age. And yet you are, as a leader in the field, you're kind of famous for being skeptical of the proposed solutions that some of your colleagues in gerontology have boldly proclaimed will make us all super centenarians. Why is it not that easy to just identify these costs that we pay in old age and just eliminate them?
Okay, so in principle, I'm a complete optimist about the radical extension of human lifespan, including human health span. Because for almost 50 years, I've earned my living extending the lifespans of fruit flies in my laboratory, not just by evolutionary genetics, but by other means, such as manipulating their diets, manipulating their access to proteins and calories, changing which qualitative kind of diet they have. In the course of my career, lifespan has been radically manipulable, and it is no big deal in my lab to take lifespan up and then take it down and then take it back up again. It's just completely malleable. Let me say that when I first started doing this in 1977, nobody around me thought that that was possible. My own doctoral advisors didn't believe in such things. So I did my first evolution of longer-lived Drosophila experiments without telling them.
That's a great story later, right?
Yeah. Well,
after successfully completing it, you say, hey, it turns out it works.
Well, I mean, I did those experiments on top of other experiments that were clinically insane. Like this was while I was also doing a 420-day quantitative genetics experiment, where I worked for 420 days straight. And then, I figured since I was always in the lab anyway, I could do this other experiment at the same time, which took my average working day up from 8 to 16 hours a day to 10 to 24 hours a day, which is how graduate students are supposed to live, right? And that has still been true of my own graduate students right up to the present day. There's nothing about evolution, there's nothing about genetics, there's nothing about biochemistry or cell biology or molecular biology that prevents the production of, in fact, immortal animals and plants. And they already exist. Hello? Trembling aspen stands, individual trees can stand there for thousands of years. Creosote bushes have been dated to more than 5,000 years old. Okay? There are vertebrates that live in the sea that live for centuries. There are clams, giant clam species that live even longer. Lifespan can be radically extended by evolution by natural selection. 40 years ago, I was given the challenge of developing proposals to do the same thing for people. And I knew that it would be insane, unethical, and impractical to propose that we evolve humans. That's the eugenics nightmare, the fevered dreams of Nazis, neo-Nazis, and fascists and racists and so on for the last 200 years or so. And no, the question was, what interventions could I come up with, given that I had a working life's worth of experience in controlling aging, that could be applied to humans? And in 84, I published one such plan. In 15 years later, in 1999, I came up with another such plan. Then about 10 years ago, I had another plan. And in the last 2.5 years, a fourth plan. And given enough resources, I think we can radically extend human lifespan. As I was saying to someone the other night, it's the classic science fiction fantasy that 3 or 4 people will come up with like the great idea, or in the case of Aubrey de Grey, one person will come up with this completely formed plan and all the technologies required to immediately solve this huge problem, like getting men on the moon and bringing them back. Jules Verne, right? And that is not remotely how powerful technologies work. Powerful technologies work when the very best scientists and technologists get together in large teams, spending large amounts of money, doing lots of pilot experiments and lots of step-by-step development. This is the American moon mission, which started off in the late 1950s. And in the early 60s, you had the Mercury program, the Gemini program, then the Apollo program. And it took a very long time, involving a lot of people and a lot of money, but it was done. Something that people had been writing fairy tales and science fiction about, going to the moon and coming back, was accomplished in 1969 in July.
So do you think that we should call people out who are selling snake oil and who are telling people that with one supplement, with one particular single lifestyle change, we can all of a sudden all become super centenarians? I won't, out of professional respect, I won't name any names who have spent their careers working on single targets. I think it is a really interesting approach to evaluate the idea of single targets, whether they're genetic single targets or molecular single targets that seem to have perhaps an inordinately large effect on lifespan, on cell regeneration, on autophagy, on senescence. But should we be actively calling out the hype surrounding these single targets and pointing, like you said, to the model of successful science programs where, whether it was the Apollo program and its predecessors or Los Alamos or any of the other massively funded, massively man-powered projects that actually pulled off what people just decades before had said was impossible, that is what it's going to take to actually crack this, not just find the magic molecule and buy it for $100 a bottle.
You're cracking me up. I have 3 answers to that challenge. The first one is almost all my colleagues, not all, but almost all my colleagues, all through the 20th and early 21st centuries, were trained to find the pathway for the biological attribute they were interested in. The gene or a handful of genes that were responsible for eye development, learning, cancer, aging. The whole weight of reductionist biological research, as they proudly called themselves, reductionists, was, let's find the gene or the key pathway, like telomere attrition, that is the cause of aging. We'll solve that problem. We're done. Okay? In fairness to them, this is an absolutely valid model for genetic diseases, which are significant contributors to human misery in between 3 and 8% of people. Okay.
Right. And that number, we have to let that number sink in. That whole single nucleotide polymorphism, one problem, one missed nucleotide or one substituted nucleotide leads to disease. This blueprint, solve the typo in the blueprint promise that the NIH still has on the Human Genome Project website to this day. It is great, like you said, for less than 10% of the human disease burden. And why did that not work out for the rest of us? Why didn't that work out for aging either?
Okay, my second response. So we looked at how evolution by natural selection produces much longer lived organisms that have extended health span, extended functionality, extended reproduction, extended and enhanced everything all through adulthood, which is what enables them to live longer. We looked at the physiology of what was involved. Not everything was radically enhanced, but a lot of things were radically enhanced. We looked at the genome. How many sites were statistically very strongly changed? That is to say, how many locations across a fruit fly genome, which is much smaller than ours, had to be transformed to double lifespan? Around 500. How many transcripts had to be changed in abundance substantially? 500 to 700. How many interactions are there between those 500 genetic sites and 700 transcripts? Thousands, because one genomic change produced changes in 5 to 8 transcripts, and each transcript change was affected by like 8 to 10 genetic differences. How many metabolites were changed? Over hundreds, thousands. We published the genomic and transcriptomic results fully. To produce these radical transformations in lifespan requires retuning the genome at hundreds of sites, retuning the transcriptional spectrum for hundreds of transcripts, metabolites, thousands. Is that simple? No. Are there one or two things of overwhelmingly large effect? No, because we went and estimated them.
This is kind of worth repeating, that the idea that we treat the body like a machine, if a car starts to have an oil leak and the engine is smoking, and we say, hey, there's the oil leak, we have a fairly reasonable assumption that is borne out by mechanical intervention that we can find the problem, we can fix the problem, and to some degree, we can get the machine running and continuing to function through this sort of seeking a single target and mechanically repairing it. You guys did not. You didn't go in, to be clear for the non-biologist listener, you didn't go in and change 500 sites manually by the mechanical assumption that the bodies of these organisms are machines. Okay. You let evolution do it, and then you found that it did not match the assumptions of the reductionists.
You have set up my third response to your original challenge. How do you make a car that's going to last twice as long with twice the cargo carrying capacity or twice the acceleration? You change hundreds of parts out of thousands. You don't change one part. You even want to make a machine do radically better. You have to change it.
Not talking about a single point of failure. If we're talking about something like why a car that was built in 1905 is driving down the road in a vintage car show, that is the case. They are typically almost fully rebuilt with some sort of replica components. But this kind of gets to this idea that when we have something that's undergoing extensive distributed mechanical wear and tear, distributed molecular disruptions, the single target intervention is essentially a dead approach right out of the gate.
Living things are very complex bits of machinery built by evolution by natural selection. Think of how many parts did you have to change to go from a Ford Model T in 1920 to a Honda Accord in 1990. You had to change hundreds and hundreds of parts. Because living longer is not a question of fixing one problem. It's a question of improving hundreds of functions, okay? And to do so harmoniously. So in fact, engineers do supply useful guidance on our project. How many things had to be invented? How many things had to be improved to go from Goddard's rockets of the 1930s and 40s to the Saturn V that took the Apollo mission to the moon and enabled the astronauts to get back alive and healthy? Thousands of things had to be done. Let's be effing realistic. Yeah, I'm all for producing people that live 2, 3, and 4 times longer with long preservation of faculties and functions. Do I think that retuning any one single pathway will get us there? No. Do I think it's impossible? No. Do I think it's hard? Yes. Do I think it's doable? Yes. How will it be done? By teams that are large, that are not kidding themselves or anyone else about it being easy or simple.
So you're not going to have somebody who wakes up tomorrow and says, hey, we've discovered a way to make the cell membrane last for a long time without solving the problem with mitochondria accumulating oxidative damage. You're not going to have somebody who solves the mitochondrial oxidative damage problem who doesn't simultaneously extend the somatic replication fidelity for somatic genetic material. It's literally hundreds and hundreds of problems. And the only realistic way to solve aging is to solve all the problems at once.
Let me administer the final coup de grace to the reductionists. The much longer lived fruit flies, living twice as long, either have no change in molecular genetic functions that are of such interest to molecular genetic reductionists, or they have higher rates of damage than in their shorter lived compatriots and ancestors. Okay. They actually accumulate damage at a similar or higher rate. Goodbye,
fascinating
reductionists. That result is so counterintuitive that when it was obtained by molecular reductionist colleagues of mine in the 1990s, they refused to publish it. I said, we'll do the entire experiment over again, massive replication, and will you agree to publish it if we get the same result again? They said yes. We did the whole experiment over again, massive replication, same result. Would they finally agree to publish it? No.
Because it breaks our expectations that this is, in fact, the route to making something live longer.
The human mind loves stories. Before Galileo, astronomy and astrology were thoroughly confused. I would submit to you that biology before Craig Venter was still in its astrology phase, where we loved the simple stories. Very occasionally, they were true. I'll give you another bullet, one in the head, another in the chest, 2 bullets, to the reductionist theories, which we published in 2012. Because we did the work, we got to publish it. If you stack up the 100 plus longevity genes that were published in the literature circa 2010, when we published our first genome-wide unprejudiced look at the sites of genetic change required to double lifespan in fruit flies. Longevity genes for fruit flies, genome-wide analysis of the experimental evolution of longer lifespan. And we compared the 100-ish longevity genes with 100-ish randomly selected sites in the genome against the genomic reference for successful doubling of fly lifespan. The random selection of genetic sites did better than the longevity genes created by my reductionist colleagues earlier, and entirely separately. The lab of Jim Kurtzinger looked at individual such longevity assurance genes, and they found the results were irreproducible. Irreproducibility is not just a problem in the David Sinclair lab. It's a characteristic problem in molecular genetic studies of aging. For that matter, it's a characteristic problem of reductionist biomedical research as a whole, which has an appalling reproducibility result. Our experiments and our flies, eminently reproducible. Anybody in the world can do our experiments, and they do. Anybody in the world can look at the genomes of their much longer lived organisms. And when they do so with enough statistical power, they find many sites in the genome play a role.
This has also been borne out in non-laboratory settings as well. When we compare the genome of a relatively short-lived whale to a very close clade with an organism like a bowhead whale, the discovery with the bowhead whale genomic analysis was not, hey, look, it found this one target longevity gene and it duplicated it a couple times or it upregulated it. It was massive changes across the genome at a ton of sites, including many, many unexpected sites. Is it fair to say that there aren't sites in the genome that can escape the way that aging is built in? Or are there islands within the genome that really truly do not play a role in aging, or are there hot spots? Is there any sort of method to the madness of the distribution?
Okay, so remember, we found 500 sites in the genome, not the 14,000 that correspond to the 14,000 genes in a fruit fly genome. And actually, a number of the 500 were not in genes. They were away from genes. So if you think of it that way, there are like, 50,000 separable sites in a fruit fly genome, and we're getting change on the order of 1% to produce a doubling of lifespan. I don't think you have to change absolutely everything. In that sense, I think radically altering a genome to produce a longer-lived organism is actually easier than going from a Model T to a Honda Accord.
So how do we go about this? You've talked in recent years about your excitement for the integration of what was once futuristic AI and is now perhaps on the edge of reality. How do we bring on the massive manpower and perhaps the added brain power of AI to solve this? How do we get from where we are, realizing that the reductionistic single target method is not a realistic model, to this future where we can say these are the sites in the genome that we need to look at?
Okay, so in addition to AI, of course, we've undergone the genomic revolution over the last 25 years, right? So we have greater power to look at all the sites in the genome, all the transcripts, and a very large fraction of all the metabolites, including proteins, in the body of any animal we happen to be interested in, especially humans, which is the most studied animal from the standpoint of modern multi-level omic research. And we are in the process of accumulating massive amounts of human data, combining genomics, transcriptomics, metabolomics with clinical data. It's all there. One example of such research is GWAS, genome-wide association studies. And what does GWAS tell us about? How many sites in the genome contribute to almost every character of interest? Hundreds.
Yeah, massive pleiotropy to some extent, except that a lot of GWAS studies for diseases we really care about come back and tell us, hey, the heritability seems to be way lower than expected.
That's because they don't have all the sites. Okay. So, I mean, in the lab with fruit flies, we can approach saturation on the number of sites, so we can get very high level of predictability from what we do. So that's primarily a scale problem. And it's also a signal problem because in the lab with the fruit flies, we have much bigger signals than exist in human populations, either within or between, because we are a little differentiated species. There's not very much difference between any outbreak human population, very small differences. We have vastly greater differences in my lab. But here's a third factor that makes me optimistic on top of omics and AI, and that is the fact that about 80% of fly genes are in humans doing pretty much the same things. And when we have looked going from our fly genomics to human genomics, it's an even higher proportion of the genes involved in retuning aging in flies are implicated in human diseases and functions.
Fascinating.
We have a first draft. And in fairness to all of my colleagues, that's nothing new about this, because for 100 years more, people who've done work with fruit flies, like Rolf Bodmer on Tin Man, have turned around and found the same gene doing similar things in humans. And since our research indicates that the kinds of genes that retune aging in fruit flies are ordinary, everyday housekeeping genes that also exist in humans, we do get some first draft information from fruit flies.
Well, I'm excited to see where the omics takes us, where the AI takes us, and where this more informed evolutionary population genetics approach to aging takes us. I hope everybody who is listening, who is watching, takes the time to follow up with some of Dr. Rose's amazing books, especially his most recent book. I will go ahead and put a link for that. And I've enjoyed your writing. I have to say, if anybody is looking for some fun reading, I just have to include a quote from your book, The Long Tomorrow. I love how you shared the story of the Russian biologist Elie Mechnikov, who won a Nobel Prize quite some time ago. And he had theorized that battles with intestinal bacteria were responsible for aging. And I love your colorful description that it was a, quote, classic example of an attractive theory killed by an ugly fact. So you have a very fun and vivacious writing style that really kind of brings to light some of these fairly advanced science topics and helps us see beyond some of the fluff that's out there, because aging is definitely a space that is ripe for fluff and sensationalized claims. It's always fun to hear the fundamentals of math-based science for how we can actually tackle this age-old problem of human aging. Thank you so much for talking with us, Dr. Rose.
Absolute pleasure to be with you.